Okay, got it. Thank you. All right. Good morning, everyone. Welcome to our monthly lecture series in high field MR at the Kirby Center at Johns Hopkins and the Kennedy Creek Institute. So this month, our speaker is Dr. Susie Huang. Dr. Huang is a neuroradiologist, and she also serves as the director of translational MR imaging at the Martinez Center in the radiology department at the Mass General Hospital. So her research focuses on the development and translation of novel MR imaging techniques for neuroimaging, and as we all know, she's an expert in high gradient diffusion MR acquisition and analysis, and currently she's leading the effort to develop the next generation ultra high gradient Connectome MR scanner for human brain imaging. And also Susie has had lots of contribution in areas such as fast neuroimaging methods and their application for clinical use. So I think the title of our lecture series is called High Field MRI, MR. But I think in terms of a diffusion MRI, there's still considerable debate whether it's more important to have a high field or high gradient, or perhaps both. So in regard to this, I think perhaps Susie is the best person to tell us what she thinks, along with many other very innovative work she has been doing, especially in diffusion MRI. So without further ado, Susie, looking forward to your talk.
Oh, thank you so much, June, and thank you everyone for the invitation to come present here today. I wish I could be there in person with you all. But yeah, when June invited me to give this talk, at first I was like, well, it's about high gradient fields. Yeah, I guess in follow up to this ultra high field workshop in Lisbon that June was on the organizing committee for, I did do like a pretty thorough exploration of the benefits of ultra high field for diffusion MRI, but essentially basically came back to the same conclusion, is that right now, I think that the part of the hardware that really benefits diffusion MRI is the gradients. And so today I'll be focusing on sort of how we can map tissue microstructure in the human brain using high gradient diffusion MRI. So I'm going to really just sort of live in the space that I do right now. But yeah, just to make note that there are actually really exciting developments that are happening in terms of integrating high performance gradients with both actually ultra high and ultra low field scanners. And so that'll be very interesting to sort of explore in the future. So as June said, my research sits at this interface of technology development and clinical translation with a focus on high gradient diffusion MRI. And today I'll be providing an overview of our work on developing, validating, and translating high gradient diffusion MRI technology for probing axonal integrity and cortical microstructure in the living human brain. So I wanted to start with taking a step back in time and to connect our current efforts in sort of MRI technology development with the underpinnings of modern neuroscience, to really get back at the heart of what is it that we want to see. So neuroscience owes a great debt to Santiago Ramon y Cajal for his landmark description of the cellular underpinnings of neural structures and their connectivity. And the essence of what he saw is depicted in this diagram. So, and as well as many other drawings that he made. And so that essence is that the nerve cells in the human brain have wires. So they're dendrites that receive information and axons that transmit information. And so his big idea was that the basic unit of neural processing in the brain was this sort of axonal cell body dendrite unit. And this insight that nerve cells are functionally interconnected with the axons transmitting that information and then interfacing with other cell bodies as the receivers, it's really been appreciated through this groundwork that neural connectivity holds the key to brain function. It turns out that through many decades of work since these initial drawings, that this basic foundation laid by Cajal is actually a gross oversimplification of what actually happens in not even the human brain, but also just in basic organisms. So this is a volumetric electron microscopy reconstruction of the fly optic lobe. And you can see even from this very small volume of tissue that the cells and axons within this small neural processing unit are actually quite complex. They're very, there's many more cell types and types of connections that are actually present than what can be gleaned from a simple diagram like this. And so that actually kind of poses one of the problems, I think, that we're trying to address now with our projects. The other one is being that a necessary sort of foundation and challenge for human systems neuroscience is defining that complete connectome, the matrix of connections between different nodes of the nervous system on multiple scales. And this is one of the biggest challenges, in fact, in mapping human brain structure. So if we believe that if we can understanding human brain function really relies on knowing its structure, then we need to be able to bridge these scales. Tools like MRI offer the spatial range and resolution to image the whole brain, so down to maybe 1 millimeter cubed voxels. And on the other end of the size spectrum, electron microscopy and other microscopic imaging tools can render those connections between neurons, axons, and dendrites with voxel sizes of less than 100 nanometers cubed. So you can see that the size scales in the human brain really span at least 4 to 5 orders of magnitude in size, and it becomes a sort of enormous challenge in terms of mapping human brain structure. So how do we get at sort of tackling this challenge? Well, one approach that we're taking here is that sort of using diffusion MRI. So diffusion MRI is a unique tool that provides an indirect probe of structures on the micron level in tissue, using the Brownian motion of water to infer, say, the sizes, cells, sort of sizes of cells, the packing density of axons. The dimensions of these sort of structures on the order of microns are much smaller than the nominal voxel size of MRI, which is typically on the order of millimeters. So as such, I would sort of venture to say that diffusion MRI holds the most promise among non-invasive imaging methods for probing cellular structures of any depth and location in the human brain. And this has led to this central vision in tissue microstructure imaging of offering in vivo histology. That is the ability to estimate and map histological features of tissue using diffusion MRI. And so if we recognize that diffusion MRI, improving the sensitivity of diffusion MRI will ultimately allow us to get at such microscopic measures, we want to sort of investigate, well, what aspects of the system will allow us to gain better sensitivity to these features. Here at MGH, we've taken sort of on the challenge of developing MRI scanners with very strong gradients to improve the sensitivity of diffusion MRI measurements. So why do we need strong gradients? Well, in a conventional MRI system, with a Gmax of typically 40 to 60 millitesla per meter, in order to achieve strong diffusion weighting, these diffusion encoding gradients must be played out for a long time. And this really limits the minimum diffusion time that can be sort of achieved and pushes out the signal acquisition to long echo times, thereby sort of diminishing SNR due to T2 relaxation related signal decay. So about 10 years ago, we embarked on developing sort of a very strong diffusion gradient machine known as the Connectome system. This was done in partnership with Siemens as part of the original human Connectome project. And in this Connectome system, this is a human MRI scanner that is equipped with maximum gradient strengths up to 300 millitesla per meter. This is about sort of 6 to 7 times higher than what you see on a conventional scanner. So using these strong gradients, we can achieve a given diffusion encoding area in much less time. And this in turn reduces the entire diffusion encoding period and the echo time. So one of the most straightforward benefits of having high gradient strengths is to basically improve signal to noise ratio by allowing us to achieve shorter echo times. But there are other benefits to having strong gradients. So the larger gradient amplitudes also enable us to do stronger diffusion encoding. That means larger sort of diffusion encoding gradient areas using these much higher amplitudes. And this can be achieved in practical times. So this really the Gmax, you can think about it as being related to the diffusion resolution. So the higher the maximum gradient strength, the better the resolution of our diffusion measurements. That is the minimum distance that can, water displacement distance that can be detected, and will be smaller with a higher gradient strength. And then finally, the ability to get to shorter diffusion times enables us to map out the water probability distribution function, that diffusion probability density function much more sharply, and will allow us to, say, resolve secondary and even tertiary fibers for, say, diffusion tractography better. So the original Connectome effort was performed by our team here at MGH in partnership with Siemens back around 2011 or so. And the centerpiece of this project really was the design of this whole body scanner with a maximum gradient strength of 300 millitesla per meter per axis, up to 200 tesla per meter per second of slew rate. In addition to having this sort of improved hardware, we sought to enhance the efficiency of the diffusion sequence to enable whole brain diffusion imaging. So we implemented novel sequence developments such as simultaneous multi-slice imaging, which allowed us to roughly triple the encoding speed by exciting multiple slices at a time and then unalias those simultaneously to enable whole brain readouts with shorter repetition times and shorter echo times. And to augment this accelerated imaging approach, we also developed a high sensitivity, a 64-channel tight-fitting brain array that allows for very high sensitivity brain imaging. So this whole constellation of technical advances achieved nearly 10 times increase in sensitivity for high B-value diffusion MRI. And to give you a sense of just how much sort of power is actually going into this machine when it's operating at maximum gradient strength, we have 4 gradient amplifiers along each of 3 axes, and those are each being fed by 900 amps of current. So in total, if you play this out, the gradients out at maximum gradient strength, it's about 10,800 amps running at 2000 volts. It's about 22 megawatts of power. And I think borrowing from one of the analogies Larry Wald made, it's actually equivalent to the amount of power that's needed to run a Los Angeles class nuclear submarine. Sort of closer to home, we're located out here in Charlestown in the Navy Yard, which is right adjacent to the part of Boston where MGH is. So our research campus is here, and we were sort of estimating that when those gradients are played out at maximum gradient strength, it takes about a quarter of the power from the small town of Charlestown to run this. So why do we need such high gradient strength, and what was the goal of using these, especially for the first Connectome project? So the goal of the original human project, Connectome project, was to map the macroscopic structural connections of the living adult human brain using diffusion tractography. And so our part of that human Connectome project was really designed to validate the improvements in signal to noise ratio using these high gradient strengths to achieve higher B values for diffusion tractography, and to validate the improved resolution of white matter structural connections in the human brain. And then as an important sort of product of this project, we provided and shared a data set of 35 healthy subjects that could serve as gold standard data to inform other diffusion MR studies using lower B values, such as those that were being performed by the WashU Minnesota Consortium. So if we take that the original Connectome project was really sort of meant to map out all the structural connections in the human brain, we also recognize that an important byproduct of this Connectome sort of technology development was that we could extract a multitude of tissue microstructural parameters with better sensitivity using those high gradient strengths. And these kinds of biophysical parameters include, say, the relative volume fractions of restricted water within axons and then hindered water in the extraaxonal space. If we use the strong gradients, we sort of recognize that we can start to sensitize the diffusion MR measurement to water inside axons, inside these very small restricted compartments, and get an estimation of compartment size, such as axon diameter. And then finally, those strong gradients, as was used in the original human Connectome project, was used to sort of demonstrate that we could get better delineation of the orientation distribution of water, say running within the axonal fiber bundles. So one of the earliest demonstrations that we did in our group to sort of demonstrate the benefits of higher gradient strengths for mapping tissue microstructure was to show how these high gradient strengths could improve estimates of axon diameter and density in healthy individuals. So here I'm showing maps of axon diameter and density that were estimated in the midline corpus callosum in a healthy person using different maximum gradient strengths. And so we used up to the maximum gradient strength on the Connectome scanner and then went down systematically down to sort of roughly state-of-the-art research scanner level, like a Siemens Prisma scanner, or even I think Philips Achieva level. And so we saw that increasing the maximum gradient strength really did improve the estimation of axon diameter and density, as well as the sort of, which can be seen through the sort of spatial coherence of these maps. And so higher gradient strengths really do allow for improved diffusion resolution of small structures. And these results show the importance of being able to use high gradient strengths and access these high gradient strengths for in vivo axon diameter estimation in humans. And of note, I think these results have been reproduced by other groups that have now sort of installed Connectome scanners worldwide, including the scanner out in Cardiff, led by Derek Jones and others. And then there's another one in Leipzig. And so these sorts of results are very much consistent when you look at them across different scanners of the same type. So now that we have this sort of very strong gradient system, and we've sort of demonstrated its ability to map microstructure, sort of getting at in vivo microscopy, I was very interested in sort of exploring its role in helping us probe axonal pathology in a variety of diseases. And one of those diseases that is probably very sort of could benefit from this kind of technology is multiple sclerosis. So multiple sclerosis is more widely known as an inflammatory demyelinating disease. And while demyelination is considered the hallmark of MS pathology, the resulting axonal damage is thought to be the pathologic substrate for clinical disability. So essentially damage to the axons over repeated episodes of demyelination ultimately kind of leads to the neurodegenerative component. Of note, the imaging correlates that are available for axonal damage are actually quite limited for MS. So a common sort of clinical dilemma that we see is that patients with MS, it's very easy to identify these sort of demyelinating lesions on a FLAIR image. And when you image patients over time, they may sort of accumulate lesions, especially in the more inflammatory phases of the disease. And then for like the progressive phase, you'll see that the lesion burden doesn't change, but the brain volume starts to shrink. And it's thought that that sort of axonal damage is really leading to that underlying axonal damage. It's invisible, but it sort of manifests in this sort of loss of white matter and gray matter volume. So overall, there is a need for improved biomarkers of axonal damage that would give evidence to the neuroprotective effect of MS treatments, especially the ones that are coming online for neuroprotection. So in terms of what's available right now for diffusion sort of imaging in terms of clinical and research studies, the diffusion tensor model has been widely used. It is the simplest yet very powerful way of capturing alterations in axonal structure. And in MS, we know that generally, as in many diseases, the fractional anisotropy of the white matter will decrease and the mean diffusivity will increase, especially in the presence of lesions. If we look at other DTI metrics, such as axial diffusivity, these changes in axial diffusivity are thought to reflect axonal integrity. And the radial diffusivity is thought to increase in the presence of demyelination, essentially sort of losing the barrier to diffusion perpendicular to the axon. As it turns out, through a variety of radiologic pathologic studies, these DTI measures, while they're sensitive to demyelination and axonal loss, they're not entirely specific to those processes. So this was a really nice RADPath study that was done at WashU by my collaborator, Eric Clowater, who's now a neurologist here at MGH. And in this study, they compared systematically the DTI metrics with histochemical staining for demyelination and axonal damage in 9 postmortem spinal cord specimens taken from MS patients, and then 5 healthy sort of controls, sort of non-neurologic controls. And what they saw here was they delineated ROIs in areas of demyelination as well as axonal loss, and then looked at the sort of DTI measures in these areas. And what they saw was that the radial diffusivity can serve as a surrogate for demyelination, that it does in fact increase in the presence of demyelination, but that it also increases in the presence of axonal loss. And so even radial diffusivity is not really that pathologically specific within this chronic human MS tissue. So putting two and two together, what we sought to do was to see, okay, can we see signatures of axonal damage, sort of changes to the axonal morphology in living MS patients as they're experiencing sort of the disease. And so we did this pilot study together with Dr. Clowder and colleagues, where we imaged 6 MS patients. We looked at the midline corpus callosum of these patients. All of these patients of note had white matter lesions within the corpus callosum, which is a commonly affected white matter tract in MS. And we mapped out the axon diameter and density using high gradient diffusion MRI. And what we saw was that in areas with lesions, there was an overall increase in the axon diameter and a decrease in the axon density. You could see that highlighted in a few of these subjects here. And these results were really intriguing because they actually are very much in keeping with findings on histopathology that show evidence of axonal dropout and also enlargement of axons within MS lesions. And so here I'm showing you sort of histological sections of axons in normal appearing white matter and in sort of MS spinal cord. And actually this is a normal control and this is an MS patient. These were stained with a silver stain for axons. And on the MS cross-section, you can clearly see that these axons appear enlarged and there's overall fewer axons in the cross-section compared to the normal non-neurologic control. And so it's not entirely clear sort of what the process driving this sort of observation of enlarged axons is. Could it be that smaller diameter axons are preferentially affected and it's the larger axons that are remaining? Could there be some sort of compensatory hypertrophy and preference or selection toward the larger diameter axons to preserve sort of conduction? So this is something that we're actively exploring through systematic sort of histologic, pathologic, and then in vivo studies now on our Connectome scanner. And so we sought to sort of validate these findings that we had in the smaller group of pilot subjects to a larger cohort. So here we evaluated the axon diameter and density obtained by high gradient diffusion MRI in the normal appearing corpus callosum of a larger group of MS patients and compared them to healthy controls. We also sought to evaluate the sort of relationship of these advanced diffusion MR metrics with disability. And so in this group of patients, it was 30 MS patients, 23 healthy volunteers. What we saw was that, again, there is an overall increase, a significant increase in axon diameter and decrease in axon density in the MS patients compared to the healthy controls. And that this trend is similarly paralleled in the progressive MS patients compared to the relapsing remitting MS patients who have less severe disease overall. We then went to look at sort of how these axonal measures correlate with measures of physical disability as well as cognitive dysfunction mediated by the corpus callosum. And so we looked at axon diameter, axon density, and then DTI measures. And it turned out that among all of those diffusion measures, the apparent axon diameter was the strongest and most significant predictor of disability as evaluated by this expanded disability status scale. So the diameters increase with worsening disability. Cognitive dysfunction is also a very prominent feature and manifestation of MS, particularly in the later stages, but can occur quite early. And so in the patients who are undergoing evaluation with our Connectome protocol, we perform sort of a cognitive battery in all of these patients to assess their cognition. And we looked at sort of the correlation of these axonal imaging measures with specific measures of processing speed and working memory, the symbol digit modalities tests, as well as new learning and memory, the brief visual spatial memory test. Both of these tests of note sort of are tests of they rely on interhemispheric connectivity across the corpus callosum. And what we saw was that overall there is sort of a significant correlation and poor performance on these cognitive measures with increasing axon diameter. And these functions sort of indicate that they may be, that this axon diameter measure may be sensitive to the underlying substrate that is actually being affected in the MS patients that may be sort of reflected in the poor cognitive function as reflected in these specific measures. So we're now actually embarking on trying to generalize this approach to map out axonal integrity, axon diameter, axon density throughout the whole brain of MS patients. So here we've developed this method, which we call tract caliber MRI, to map axon diameters across fiber tracts throughout the whole brain, regardless of the orientation. So what we're seeing here is this is the initial sort of mapping of axon diameter across fiber tracts in the whole brain of 20 healthy volunteers. We did this at 2 millimeters, sort of isotropic spatial resolution with whole brain coverage. And even with the first few volunteers, we started to see some very interesting patterns in terms of the relative axon diameter that was emerging in certain tracts. For example, there's this very large sort of diameter tract that appears to run pretty much along the expected course of the corticospinal tracts. And these are known to be very large diameter fibers that project from primary motor and somatosensory cortex. Furthermore, we also observe this interesting anterior to posterior gradient of axon diameter with smaller diameter fibers in the frontal lobes and larger diameter fibers in the parietal and occipital lobes. And these findings are very much in agreement with what's been shown from sort of axonal tracing this is a study from non-human primates. But it actually does sort of suggest that we're able to capture some feature of the axonal morphology that agrees with what has been seen under the microscope. And that we're sensitizing our measurements, our diffusion measurements, to these very small compartments using the high gradient strengths. So what we've realized in the course of these experiments, in fact, is that in order to gain even better sensitivity to the actual distribution of axon diameters, which are actually more on the order of 1 micron as opposed to 5 or 6 microns, we need to go to higher gradient strengths to really get sensitivity to those small compartments. So toward that goal, the Connectome 2 project, which is an NIH-funded brain initiative project, it's a multi-institutional collaborative project that is really targeting the development of a next generation human connectomics and microstructure MRI scanner. And so this scanner, we're basically tailoring it to infer cellular and axonal size and morphology at unprecedented spatial and diffusion resolution throughout the whole human brain. And so we're hoping that this will provide greater sensitivity as well as imaging speed for structural imaging using diffusion MRI at multiple scales in living human subjects. So the centerpiece of our project, as with the original Connectome project, is the development of a next generation ultra high gradient system with ultra fast slew rates. So we're going to target nearly doubling the Gmax and tripling the slew rate. This was an early schematic, but this gradient coil is now being constructed in partnership with Siemens and Erlangen, and it's taking the form of a head-only gradient sort of coil design. So alongside the sort of gradient technology, we are developing advanced image reconstruction and acquisition sort of hardware and software. So this includes the construction of high sensitivity RF coils. We're going to have a sort of very high channel count in vivo and ex vivo coils, as well as integrated field camera monitoring to sort of correct for real-time eddy current distortions. And then finally, I think recognizing that all of these measures that we're doing of microstructure are indirect. Ultimately, I think what will really drive the field forward is to be able to validate the findings that we're sort of inferring from our diffusion MRI measurements and to calibrate, if you will, our diffusion MRI measurements with orthogonal imaging modalities, ground truth modalities of tissue structure. And so we're doing systematic multi-scale validation using ex vivo diffusion MRI measurements and sort of comparing those signals with what we would infer from diffusion MRI simulations in realistic tissue substrates derived from scanning electron microscopy, and then micro CT as sort of a bridging technology that goes between the very fine scales and the larger macroscopic scales. Yeah, so to clarify, why do we need a Connectome 2 scanner? Ultimately, I think just based on the physics, higher gradient strengths and slew rates are needed to achieve shorter effective diffusion times and probe these micron-sized structures. We also need, as I mentioned, systematic multi-scale validation to inform better biophysical models of brain tissue microstructure. And then finally, once we sort of achieve this sort of hardware and sort of analysis altogether, ultimately our vision is that pushing the limits of in vivo diffusion MRI will enable us to infer how brain structure and connectivity are changing across scales and behaviors. And so this applies not only to pathology, as I've demonstrated with multiple sclerosis is just one example, but ultimately what we'd like to do is to get at the microstructural basis of neuroplasticity. And so we actually have ongoing experiments in which we are training subjects in the scanner to learn different types of tasks, such as like a piano playing, keyboard task, and then looking at the diffusion measures, including those in the microstructural measures in the gray matter to see what is actually changing. And so these experiments right now are actually quite preliminary, but I'd be very excited to sort of share with you the initial findings, especially as those findings are reproduced. So in the meantime, as I mentioned, our Connectome 2 scanner is, the construction is well underway. We're targeting very high maximum gradient strength of 500 millitesla per meter and slew rates up to 600 Tesla per meter per second. And so a lot of our hardware development around the scanner is modeled after our original and our experience with the original human Connectome project. So in the original human Connectome project, we actually had ex-vivo brain sort of slab coils that would allow us to get diffusion MRI measurements in sort of thick sections of tissue. And we're now sort of converting that to get very high sensitivity whole brain ex-vivo imaging. And then likewise for the 64 channel head coil, we're now sort of extending that field of view. We're going to include portions of the sort of brainstem, the parts of the neck that we can fit within this head only gradient. And then finally, an important component is this field camera for real-time eddy current correction, which I'll show in a few slides. So beyond the hardware and moving beyond tractography from our original Connectome project, we are also implementing a variety of advanced diffusion encoding experiments for probing tissue structure at multiple scales in the cortex, specifically looking at the gray matter, looking at measures of cellular size and morphology. So we're moving beyond sort of our standard canonical statistical Tanner encoding with the pulse radiant spin echo experiment, which we know will benefit from these high gradient strengths for measures of, say, compartment size in axons and cells. So we're moving beyond that, and we're now implementing sort of oscillating gradient spin echo experiments, which I know actually I was really inspired by the work that Manisha Agarwal, Susie Mori did with the OGSC experiments in the cerebellum ex-vivo on your small bore scanners. And we're designing experiments that will sort of achieve similar effects to get to even shorter effective diffusion times to, again, map these sorts of quantities. And then finally, we are actively sort of performing experiments using double pulse field gradient and multiple pulse field gradient experiments, essentially incorporating at least 2 diffusion encoding periods to get at measures such as microscopic and anisotropy and compartment shape anisotropy of the cortex. And so using these sorts of double and multiple pulse field gradient encodings, we can actually sensitize the diffusion MRI measurement not only to oriented structures at the macroscopic level, such as in the white matter tracts, but also to distinguish structures that are anisotropic on a microscopic scale. So such as these ellipsoidal cells, we can actually, or dendritic processes, we could distinguish between areas that are more structured versus more disordered, as compared to substrates that are microscopically and macroscopically isotropically, like these spherical pores here. So toward this goal of mapping microscopic anisotropy across the living human brain, we are performing these double diffusion encoding experiments, which are providing some insights into sort of microscopic domains that are distinct, say in certain cortical regions here in the perirolandic cortex, as shown by these microscopic anisotropy maps rendered on this cortical surface here. And so to try to link up some of our efforts with sort of the in vivo measurements to ex vivo validation, we're embarking on calibration of these diffusion MR measurements with multi-scale validation studies. So we have a component of the project that's focused on looking at ground truth phantoms made out of various textile fibers to sort of serve as references and validation. So if we know the compartment size and we know the configuration of that compartment, we can really reliably predict what the diffusion signal will look like there and then compare against our measurements. And then moving into more complex substrates such as actual human cortex and subcortical white matter. So this part of the project we're doing in collaboration with Jeff Lichtman at Harvard. He's a cell biologist who has really dedicated his life to understanding how connectomics within sort of not just the human, but also across multiple species. And he was able to actually get a very high resolution scanning electron microscopy data set from human cortex. This was obtained from a patient who was undergoing surgery at MGH for epilepsy. Our neuropathologist extracted a portion of tissue from the middle temporal gyrus, an area that was unaffected in that patient. And Jeff and his team have been working on this data for the last 5-6 years. And so they're basically imaging this tissue on an MRI voxel scale. The size is about 2 to 3 millimeters, so it does approach what we can see on an MRI with very high sort of electron microscopic resolution. And so the challenge of dealing with such large volumes of tissue on the microscopic scale is just the sheer amount of data that is generated. And so as part of our approaches to validation, we started by looking at the subcortical white matter, so this corner of the tissue here. And we've actually worked with Jeff and his team and also ourselves segmented volumes of white matter tissue from the specimen. And so we can actually get sort of pretty complete morphological segmentation of the axons, as well as the axons plus the myelin to give us sort of realistic tissue substrates that we can then perform Monte Carlo simulations within to really simulate what does the diffusion MRI signal look like in this really complex agglomeration of axons. And so with this kind of substrate, what we're seeing is that the axons, in fact, are not simple cylinders as one typically models them, even with the sort of axon diameter mapping, but they're actually much more complex. The axons, they have these beating sort of patterns, and they also do kind of undulate maybe on a larger sort of size scale. So Hongxi Li is now an instructor here at MGH, and we've been collaborating with Els Veermans, Dmitry Novikov at NYU to really look at how the features, the morphological features of these realistic axons impact the diffusion MRI measurement, particularly looking at the diffusion time dependence of the signal within the realistic axons, and then sort of simulated in synthetic substrates where we take out certain portions and see how that impacts the diffusion MRI signal. And our goal here is really to get a better understanding of which axonal morphologic features will our scanner and our measurements be sensitive to, so that we can really try to validate the measurements of, say, axon diameter or axonal beating that we're getting from the ex vivo and eventually in vivo experiments. So finally, to talk a bit about the ex vivo diffusion MRI, the reason that we've built this in as an integral part of the project is that we see it as a key bridging technology for validation. So it has the obvious benefit that you can get higher spatial resolution than in vivo diffusion MRI, simply because of time. There's no time constraints. We can use specialized pulse sequences, including 3D volumetric encoding to get very high spatial resolution. And the consequence of this is that we can really try to map mesoscopic structure with unprecedented detail, maybe down even to sort of 400, 500 micron level compared to the in vivo acquisitions, where we're limited more to the sort of 1 to 2 millimeter size. So for our ex vivo validation efforts, we are leveraging state-of-the-art technology that we have in-house here, and that will be sort of seamlessly transitioned over to the next sort of generation scanner. So currently on our Connectome scanner, we have a custom-made 48 channel receive array coil for whole brain ex vivo imaging, and it really provides some very nice, very high sensitivity, particularly on the cortex. And so we're using this in combination with a 3D multi-shot EPI sequence to get ex vivo whole brain diffusion MRI at very high spatial resolution, down to here we demonstrated down to 550 microns cubed. This was presented at ISMRM last year by our team. We can get up to B values of 10,000, which is actually not that high for ex vivo diffusion MRI when you factor in the reduced diffusivity of fixed tissues. And I think one of the sort of issues that we realize in the course of doing these experiments is that the eddy currents from the diffusion encoding gradients become non-negligible and may start to affect the image readout by creating a time and spatially dependent perturbation phase. And so our postdoc, Gabriel Ramos-Jordan, has been working on this problem intensively over the last year to see if we can try to account for these nonlinear, spatially and sort of temporally varying phase variations into the image reconstruction to really get at more accurate sort of images and better image quality. And so the real impact of sort of this type of nonlinear phase evolution is that it results in very severe ghosting in the diffusion MRI measurements. And so our approach to correcting this is actually to measure the background sort of phase, the changes in the magnetic field on both the spatial as well as the temporal variations using a dedicated field camera. So this is a field camera that we had installed last year around this time, delivered by Scope Technologies, and we're actually using this Phantom, this field camera to do real-time eddy current correction on the ex vivo measurements. And the plan is really to, for the next generation Connectome scanners, to integrate field camera probes into our in vivo coils as well as the ex vivo coils so that we can do simultaneous field camera monitoring. So to give you sort of a flavor and hopefully preview of what's to come, especially we'll be presenting this at ISMRM, when we don't account for these sort of spatially varying magnetic fields, if we just sort of assume a linear model, which is typically what's done with the standard online sort of image reconstruction systems, you'll see that there's actually very profound residual ghosting in the diffusion weighted images. And you can kind of see that even more clearly when you window very narrowly. Whereas using our higher order image reconstruction with the information from the spatiotemporal field camera monitoring, much of that ghosting actually can be sort of accounted for or fixed through accounting for these spatiotemporal variations in the background field. And so this is important because it actually does, if you don't account for those artifacts, it will, of course, obviously, I think, impact the downstream diffusion MRI analyses. We actually saw this very early on with our original Connectome project. I think one of the first experiments that we, people in our team, Jennifer McNabb, Brian Edlow, these are people who really wanted to do ex vivo whole brain diffusion MRI for looking at entities like traumatic brain injury. They were, their images were plagued by artifacts like this, where you would get sort of biases in the mean, sort of in the color FA and also in the mean diffusivity simply due to this profound ghosting. So with the higher order image reconstruction, with the field camera monitoring, we're able to reconstruct much more high fidelity sort of maps of mean diffusivity, FA, and basically more advanced sort of reconstructions, advanced diffusion models. We will be able to use this data more reliably. And so the only additional point that I'll make here is that having this sort of real-time field monitoring can really be advantageous as compared to the standard approaches, which is to use sort of post-processing methods like eddy, the eddy tool from the FSL toolbox, which actually can lead to substantial loss of resolution, especially in fine scale structures like the hippocampus, where you may actually want to see those fibers going in. So when both the ghosting and the eddy current effects are accounted for in the forward model using the field camera measurements, there's really no blurring here. We can go up to B values of 50,000 or so, and you can see just how more nicely the fine structure of the hippocampus is delineated here. And so this has sort of set us up for hopefully in the long run doing ex vivo diffusion MRI, a very high spatial resolution for tractography, for delineation of fine white matter pathways, again, in the hippocampus as shown here, and ultimately to help us map cortical anisotropy. Because what we really like to see is, can we actually delineate microstructure associated with the laminar architecture of the cortex, say within different types of canonical cortices like primary sensory versus other types of cortices, moving toward this goal of potentially mapping the microstructural substrate of neuroplasticity. So I'm pretty much out of time. So I just wanted to say that all of this work would not be possible, and I haven't even highlighted all of it, but it wouldn't be possible without the collective efforts of this Connectome 2 team. This has been an amazing multi-institutional, sort of disciplinary collaboration, and has generated a lot of interesting ideas and sort of fruit for future collaborations. And I also want to thank, in particular, like my collaborators here at the MGH, as well as people in my team, for all of their contributions. And I also thank you all for your attention.
Wow, thank you so much, Susie. This is really a great talk. Lots of information, very comprehensive, talk about diffusion MRI. So if you still have time, Susie, this is the question and answer. So I see Dr. Van Zyl is showing, maybe asking some questions. And also for the rest, we are using the question and answer Q&A box, or you can simply put your question in the chat box. So Peter, do you want to ask any question?
Yeah. Well, thanks, Susie. That was really interesting. And I've been a bit out of diffusion for at least 10, 12 years, I think. But I'm getting interested in it again. And you probably can explain this in no time, but I was wondering, how can you measure axon diameter on the size of a voxel? Because you also have the myelin in there. So how do you know that what you're measuring is the axon diameter, not contaminated by the myelin?
Yeah, no, that's a great question, Peter. I have to say, like, I would love for you to get back into diffusion, because I actually refer many of your papers actually I've referred to for some of the modeling. So for our diffusion MRI measurements, we pretty much neglect, I think, the myelin right now, in the sense that it has a really short T2 time. So on the echo times that we're doing our measurements, we are not really seeing the myelin signal. And so we can be pretty confident that the sort of water diffusion that we're observing is either coming from inside the compartments or in the sort of extracellular space. And I guess the other point I would add is that the reason we're going to such high B values is that actually once you get to very high B values, the thought is that you're suppressing a lot of the freer diffusion that's maybe occurring in the extracellular space, and then getting at that very restricted water within the axons.
Yeah. And, I totally agree that you cannot, with radial diffusion, look at axon damage or even myelin damage, because they're mixed in due to similar considerations. But I always thought that when you, if you measure axial diffusivity, that you can measure the effects of axonal bulbing, because in principle, that should slow down the axial diffusion.
Yes. Yeah, and so that is something that we are, I think that, yeah, definitely you can get at it. And in some ways, actually, even in sort of the major application of diffusion MRI in clinical practice, which is this whole restricted diffusion that you see within strokes, I think one hypothesis is that the reason you see the restricted diffusion is because of potential beating of those axons, that they're sort of restricted diffusion along the length of the axon. So
yeah, thanks. Yeah, thanks a lot for the nice data. It's really beautiful. So
thanks so much. Yeah,
thanks. We got a question. Dr. Barker asked a question in the chat box, and also we got an anonymous attendee question from the Q&A box. The two questions are sort of related. I don't know if this is the same person, or probably not. But let me just read the question quickly. How is the participant experience in the Connectome scanner? Is peripheral nerve stimulation a problem and or noise? Can you comment on the biological effects of the high gradients?
Yes, yeah, so those are very relevant questions. And I have to say, this is actually work that Larry Wald has been focusing a lot of attention on. Larry Wald, as well as Matthias Davids in our physics group. So let me first kind of address this from the Connectome scanner, the current one that we have, which is a whole body scanner. So one of the concerns with the Connectome scanner and effects we were exploring even when it was installed was, peripheral nerve stimulation. So when you slew the gradients up very quickly and you go to high gradient strengths, are you hitting the peripheral nerve stimulation limit? It turns out that our scanner, because it's a whole body scanner, we've there's a gradient safety watchdog on that scanner that will actually sort of limit the amount of the slew that you can go to limit, ultimately the limiting factor is cardiac nerve stimulation, because with the whole body gradients, if you stimulate cardiac nerves, you could in theory cause cardiac arrest. It could be fatal. So that was an early consideration with the whole body design. And then the cardiac nerve stimulation limit, I think, is sort of going along with that. That is sort of addressed in the whole body scanner. Now that we're moving to this head-only design, peripheral nerve stimulation is still a consideration. And so what Matthias and Larry have done is they actually have these pretty detailed models of peripheral nerve stimulation based on whole body atlases of male and female sort of prototypes with the peripheral nerve distributions, especially kind of up in the face. And so they're working with Siemens to deliver these peripheral nerve stimulation models, and that has been integrated into the optimization of the gradient coil windings. So this was actually done earlier already as part of another high performance gradient project done under the BRAIN initiative led by David Feinberg at Berkeley with the impulse gradient. That is a high performance gradient with Gmax of 200 millitesla per meter and slew rate up to 900 Tesla per meter per second for 7 Tesla imaging. And so they kind of piloted that peripheral nerve modeling into the development of that coil. And it has, I think, proven to be quite successful. So our gradient coil for Connectome 2 is actually going to be using the same geometry as the impulse gradient coil. And we've essentially given them the peripheral nerve models, and they've iterated that to sort of adapt it for the increased gradient windings that they'll have to do to get to higher Gmax. But yeah, this is definitely a consideration. It undergoes systematic testing in healthy volunteers in the factory before the scanner is even delivered. Yeah, so those are, I think, the major considerations. There's also other things like retinal stimulation, which I could talk about, but hopefully that will not be an issue with any clinical scanners that you guys are using.
Yeah, sounds great. Thanks so much, Susie. So I feel like I'm obligated to answer to ask this question, if you don't mind.
Yeah.
So like I said in the beginning, what's your take on the possibility of combining this this high gradient system with high field system? Do you see any benefits, challenges, or other opinions?
Yes. So, I mean, as you know, June, I think my perspective on this changed during the course of the sort of 3-4 day workshop in Lisbon. I think going into the workshop, I was very much of the opinion, diffusion MRI at ultra high field, like 7 Tesla or higher, is going to be extremely challenging because you have to deal with shortening of the T2, worsened distortions from susceptibility effects. And so there are a lot of, in addition to B1, B0 and homogeneity. So there's all these challenges. I think what David Feinberg showed with his impulse gradient results was actually really kind of transformed my thinking about this. So with a very high performance gradient coupled with ultra high field, you can start to get around some of these problems because you can maybe try to outrun the T2, if you will, by using very high slew rates to do the image readout faster, and also to do diffusion encoding more effectively. And so overall, I think that that has opened up, in terms of my mind, the possibilities for doing diffusion MRI at ultra high fields. And I'm actually really curious about trying to play around with David's gradient and potentially trying to get one here on our sort of Siemens Terra scanner. Yeah, so the images you show from David during the workshop was pretty impressive. Yes. So it sounds very exciting direction in the future. Yeah. All right. Is there any other questions from the audience? If not, let's thank Dr. Huang again, and thank you all for coming to this seminar. We'll see you in a month, and thank you again, Susie. Thank you so much, June, everybody. Discussion, everything. So, thank you. Awesome. See you soon. Bye.